And so we're really excited to start with session 1, Bioengineered Vasculature, which is moderated by Dr. Alice Stanton and Saswat Chaudhry.
So we'll pass it over to the moderators now to introduce our first speaker.
Awesome. Yes, thank you so much. And thank you for all you've done to organize this. I think it's a great opportunity for everybody to come together and delve into these topics deeper and get an opportunity to get feedback on their work. So very happy to be a part of it. And I'm going to introduce the second two talks, but I'll hand it over now to my co-moderator to introduce the first two.
Thank you. I'm Saswat Chaudhry, joining from India. I'm a graduate student at the Indian Institute of Science, Bangalore, and it's a real honor and privilege to be moderating one of the sessions. So without much further ado, I'll start with the first speaker, who is Cody Crosby from the University of Texas at Austin. And he'll be talking about quantifying the vasculogenic potential of induced pluripotent stem cell derived endothelial progenitors in angiogenic hydrogels. Over to you, Cody.
Great, thank you. I'm just going to share the presentation. All right, cool. Can everyone see my screen?
Looks great.
Awesome, thank you. Cool. So I first want to start by thanking the organizers and those who put this together, as this is truly an excellent opportunity to be able to showcase our work to a very wide audience and hopefully to learn a lot from everyone else throughout this presentation. Our main goal was to build a more robust microvascular platform. So the title of my talk, as was previously mentioned, is Quantifying the Vasculogenic Potential of Induced Pluripotent Stem Cells Derived Endothelial Progenitors in Angiogenic Hydrogels. I realize that's really a mouthful at first. So our main goal was to build a quantification algorithm to look at a very specific vascular cell type that's derived from pluripotent stem cells in novel biomaterials. To cut immediately to the chase, many of us here have probably heard of in vitro vascular models, and they have a number of different advantages. The first is that they can reveal fundamental mechanisms of both vasculogenesis or angiogenesis. We can create physiologically relevant models of cardiovascular disease by isolating cells from patients with, for example, that specific pathology. And we can also screen therapeutic compounds on human vasculature. So this could be enormously helpful in avoiding, for example, the use of animal models or experiencing massive failures at the phase 3 clinical level. Broadly, in vitro vascular models can be split into 3 categories, and we'll really see those talked about today. The first is microfluidic devices, in which we can use state-of-the-art micropatterning techniques to create very precise microvasculature. We can also use 3D bioprinting by extruding hydrogels through thin nozzles. This is at least the most common method, to really control the spatiotemporal presentation of different cues in thick vascularized tissue. And most critical to my talk today is I'll be discussing the self-assembling hydrogels. This is the oldest of the 3 techniques. However, by embedding a vascular cell type in a 3-dimensional hydrogel and then exposing to a variety of physical and chemical cues, we can guide these vascular cells to assemble and to luminize vasculature. However, this is not to say, even though we've made a tremendous number of advances in vitro vascular models, this is not to say that they are without their limitations. And they really face 3 main grand challenges, so to speak. The first is cell sourcing. Many gold standard models still use HUVECs, human umbilical vein endothelial cells. These are primarily sourced, which means they often quickly go senescent in the laboratory. They're derived from a venous cell type, which isn't ideal for arterial mimicking models. And finally, most of us did not have our umbilical cords preserved at birth, which means they're probably not patient-specific for many people. With regards to quantification schema, there are 3D suites which are extremely powerful, such as Imerys, that are capable of performing such complex analysis of these vascular topologies. However, many of these software suites are prohibitively expensive, and they limit access to the source code, which is very difficult for many academic laboratories that have very custom needs. And finally, biomaterial. As we've seen, tissue engineering has made tremendous advances with the development of biomaterials over the past 20 to 25 years, as engineers and researchers seek to incorporate bioactivity so that these hydrogels are cell sensitive, and also to incorporate different material properties so that they can capture the bulk properties of tissue, which are extremely complex and extremely viscoelastic. So with regards to our laboratory specifically, we are the Zoltan Lab, so we do stem cell tissue engineering. We are fascinated by the potential of iPSC-derived endothelial progenitors to overcome several of these challenges. So 2015, which actually marked the beginning of my dissertation, these 2 protocols were released to the world. And it was found that endothelial progenitors, by using small molecules and growth factors, could be derived from pluripotent stem cells. The reason why we're convinced that these cells have tremendous therapeutic potential is they can be patient-specific, since they can be derived from pluripotent stem cells. Since iPSCs can pretty much be expanded limitlessly in the laboratory before use, we believe these are also highly proliferative in that sense. And they also are bipotent, meaning they can differentiate into the building blocks of mature functional vasculature from endothelial cells and smooth muscle cells simply by toggling between these two angiogenic growth factors. Furthermore, you can generate specific endothelial subtypes because the endothelial cells shown here are capable of differentiating into arterial, venous, or lymphatic lineages. There was a central problem that remained. Because these cell types were so new, so once again, they were discovered right about when I started my dissertation, 2015, we don't know how these cells respond to variation in the local ECM. Because this effect remained relatively unknown, this was a significant barrier to translation. So in Project 1, I'll be discussing our first objective, which was to design a novel in vitro model that contained a custom quantitative analysis to determine how ECM properties could impact the development of iPSC EPs into a vascular network. So to start, we needed to verify that these cells were actually capable of self-assembling into a 3D capillary network in a 3D biomaterial. And so what we did is we used FACS, fluorescent activated cell sorting, to isolate CD34 positive iPSC EPs from a larger differentiated cell population. So roughly anywhere from 5 to 25% of the cells were CD34 positive. So by using FACS, we then encapsulated these in collagen hydrogels. We showed pretty high viability 1 hour after encapsulation. And then over the course of 7 days, so this is day 1, day 4, and day 7, by using calcine, which simply just tracks the live cells, we were able to show that these cells slowly extend and luminize and eventually anastomose into these microvascular networks. By fixing staining and imaging on a spinning disk and focal microscope, specifically staining V-cadherin and F-actin, we were able to show that these vessels, as shown here, are luminized, anastomosed, and branched, which is what we expect to see out of a capillary plexus. Most critically, in the first project, we were able to develop a novel computational pipeline to quantify iPSC-derived capillary plexi in 3D. Specifically, we use 2 programs, which many of you are familiar with and are open source to most academic laboratories, assuming they have the MATLAB license, ImageJ and MATLAB. So in ImageJ, we performed the filtering and thresholding. So we acquired these 3D stacks on a spinning disk confocal microscope. We then did a variety of filtering techniques to remove any dead cells and spurious noise. And finally, we thresholded to create these segmented images as shown here. These segmented stacks were then transferred to MATLAB, in which we performed skeletonization using a previously developed algorithm to analyze small osteocyte networks. We then were able to trace the center lines of these vessels and convert this into a nodal graph. From a nodal graph, we're able to perform a topological analysis and look at the different numbers of branch points, endpoints, and vessels, which allows us to get a very sophisticated picture of the resulting 3D vascular networks. So as a proof of concept and really a way to look at the beginnings of mechanotransductive effects on these vascular networks, we wanted to look at the impact of overall collagen density, overall matrix density. And what we found is that as we increased collagen density from 2.2 to 3.1 to 4 mg per mil, the capillary lattice actually became smaller and less branched in these higher density hydrogels. And this fortunately was borne out by our quantitative analysis, in which we note the number of branches, vessels, and network length all decreased with increasing collagen concentration. So in Project 1, we were able to design this novel in vitro model that contained a custom quantitative analysis in ImageJ MATLAB to look at how ECM properties, specifically matrix density, impacted the development of iPSC EPs into a vascular network. Now you'll notice I use a pretty primitive biomaterial, collagen by itself, and this really was our essential limitation, as we noted that collagen compacts under extended cell culture, and it's also extremely difficult to tune without simultaneously varying other material properties. So for example, by increasing the density, we were also simultaneously increasing the stiffness, we were decreasing the pore size, we were affecting the degradability. In collagen, it's very difficult to disentangle or decouple these parameters. So we decided to use a material called NORHA. Those in the Vertic Lab will be very familiar with NORHA. In collaboration with our collaborator, Dr. Adrian Rosales at the University of Texas at Austin. And so what we were able to do very briefly is we took HA, shown here in blue, we functionalized it with Nor-binding groups, and in the presence of water, we added dithiol crosslinker, and this allowed these functionalized NORHA chains then bind to one another and form these hydrogels. The advantage of NORHA is that you can really change the mechanical properties simply by changing the concentration of this dithiol crosslinker. You can also functionalize with different ligands and dyes. Ligands to allow for cell attachment and therefore bioactivity, different dyes, for example, to measure the degradability as these dyes are released from the degrading hydrogel matrix. So we combined NORHA with the material we used earlier, type 1 collagen, to create these composite hydrogels. The main goal here of these composite hydrogels was to create these very tunable angiogenic microenvironments. So we've created something known as an interpenetrating polymer network. So the collagen and NORHA were cross-linked independently and interwoven. However, they're not chemically bound to each other, which means it's impossible to disentangle the two without breaking any bonds, but they're also not chemically interacting. So we formed an SIPN, semi-interpenetrating polymer network, by allowing the collagen to undergo fibrinogenesis for 30 minutes. We then formed a true IPN by then, in the presence of a photoinitiator, exposing this SIPN to UV light of 365 nanometers for 50 seconds. We then allowed the encapsulated CD34 positive iPSC EPs, that same cell type, to then grow for an additional 7 days in these IPN hydrogels. Now, the first thing we were able to do was decouple the stiffness from the density, as was our first goal. And so this looks like a lot at first, but I want to follow a few main trends. The first is that we were able to analyze both degradable and scrambled peptide crosslinkers. So these are more degradable IPNs, these are less degradable. As we increase the crosslink incorporation, we notice a trend with increasing storage modulus with increasing crosslink incorporation. However, we also note this is still a two-step gelation process. So the SIPN, so collagen only gelling, still forms a storage modulus of about 50 pascals, which is what we saw earlier. And in the IPN, so with the UV exposure, we notice a definitive jump at every level. Now, other groups have formed IPNs of HA and collagen. Some of those groups might even be here today. What we wanted to do is really to thoroughly characterize the collagen fibrinogenesis process, as this is very poorly studied. And what we noted were four different things, really. The first is that we noticed a lag in collagen fibril assembly. So collagen, as shown here in orange, occurs very rapidly under physiological conditions, 37 degrees Celsius. However, in the presence of HA, this lagged by about 10 minutes, as seen here. However, if we note by just simply looking at the rate constant of these sigmoidal curves, that there's actually very little difference between them. So there was a lag phase, but the rate of assembly was not very much affected. Also, there was a definitive difference in opacity between these different hydrogels. So by increasing the crosslinker incorporation, the gels did become steadily more opaque until they almost matched the opacity of pure NORHA, as seen here. And perhaps most fascinatingly, in collaboration with Dr. Salpin-Prek, we were able to perform confocal reflectance imaging to look at the individual collagen fibers here. And what we see is that in collagen alone, we have very thin, very dispersed fibers. And in the different IPNs shown here, these fibers basically bundle together. They become much thicker and much less dispersed. We also were able to update our computational pipeline. This is basically a brief summary of what I discussed earlier. You'll note that earlier we were able to analyze the connectivity and the number of vessels and branch points. In collaboration with Dr. Joshua Morgan, we were able to add the following 4 parameters. So we were able to look at overall diffusivity, so the average distance from a vessel, the volume fraction, how much is that vessel occupying of the hydrogel, vessel diameter, and the tortuosity. So basically how straight line is that given vessel. We were able to do this in both degradable and scrambled hydrogels. So first we looked at cell CD34 iPSC EP populations in degradable IPN hydrogels. And what we noticed is actually, for the most part, they tended to perform worse in the collagen. This was slightly conflated by the fact that the cells simply survived better in the collagen, which led to premature compaction of the hydrogels, which artificially increases the volume fraction and decreases the hydrogel depth. We were not able to look at anything above 50% occupancy, mainly because the pore size became so small that the cells were simply not viable. However, we noticed a very interesting trend when we switched to a scrambled sequence, in which the scrambled sequences IPNs actually perform very, very similarly to collagen until we hit 75% occupancy. At 75% cross-linking occupancy, what we notice is a dramatic drop-off in the connectivity and volume fraction parameters, which is seen here qualitatively, as the cells are single spheroids that are expanding, as opposed to an anastomosed vascular network. So, as I've hit the 12-minute mark, I want to return to the 3 main grand challenges, and that was cell sourcing, quantification, and biomaterial. So here we provide an alternate cell source, iPSC EPs, which are bipotent and can form the constituents of mature functional vasculature. We built a novel computational pipeline in both ImageJ and MATLAB to gain a better idea of very sophisticated network topologies. And finally, we built a novel IPN consisting of NORHA and collagen to better decouple, for example, stiffness from density, and to further probe such parameters as degradability in these angiogenic microenvironments. I was not able to complete this work alone. I especially want to acknowledge our collaborators, Dr. Salpin-Prek and Dr. Adrian Rosales here at UT Austin, who greatly helped us in the work that's shown here, and also the many members of the Zolden Lab, those who are currently postdocs, the graduate students, current undergraduate students who all played an instrumental role in this work. At this point, I'm happy to accept any questions, and thank you for having me.
Yeah, so we do have some questions. So the first question is, does the stiffness of collagen hydrogel has any effect on iPSC's EP?
That's a great question. I did not show this data here. We did measure the stiffness of the collagen hydrogel, but we were unable to effectively decouple it from the density. So with that results earlier, where I showed that increasing, let's see if I can back up a bit. So this was very closely correlated with stiffness because these higher density hydrogels are also stiffer, about by a factor of 3. However, it was very difficult to say which one of these was actually responsible for the phenomenon that we were observing.
Okay, and someone is asking that, are these networks perfusible?
We believe they would be perfusible. Currently, because we don't really have a way to hook them up to an inlet, we're not able to perfuse them currently, but that is one of our current aims.
Okay. And I was just wondering, like, if the nature of hydrogel would also matter, like here you have used collagen. So if you use maybe some other hydrogel, like fibrin or something else, which may be cationic or something, would that matter?
That's a great question. It's actually something we're looking into currently. It may very much affect the kind of dispersion of the fibrils as shown here. So I think we would begin to see different architectures depending on that repulsion, as you mentioned earlier.
Okay, thank you. Great talk. So we would be moving to the next speaker, who is Parinaz Fatih from National Institute of Standards and Technology and University of Illinois. Vessel on a chip with cyclic fluidic flow. Over to you.
Your talk, and I'll pass the floor to you.
Okay. So are you able to see my presentation?
We have you starting. Oh, perfect. There it is.
So thank you very much for this opportunity. And today I will present my PhD work. Actually, I'm finishing my PhD this year. And I will introduce you to this presentation entitled Biobassilonachip to probe endothelial permeability against nano-delivering system. So basically, the vascular system is composed by an intricate network of macro and micro vessels that representing one of the major organs in our body. Vasculature is responsible for the efficient exchange of oxygen, nutrient, and waste within and between our tissue. Endothelial cells that line the and vessels act as a selective barrier for immune cells, nutrient, oxygen, hormones, ions, and also, for example, drugs and nanoparticles. The exchange at the vascular barrier can be either paracellular or transcellular. However, endothelial cells display tissue-specific phenotype according to the tissue in which they reside. Like, for example, endothelial cells that are in the kidney are more fenestrated respect to the ones that are in the blood-brain barrier that are tightly connected. So in order to recapitulate the vascular system, in vitro vascular network have been fabricated and are called Organoids. And this model allowed us to reproduce the human pathological and physiological condition and also test the nanoparticle permeability and also drugs permeability in our body. In the past, co-culture of endothelial cells and supporting cells was done with the 2D planar system, like, for example, transwell system and Boyden chamber that do not recapitulate the 3D organization of vascular system. On the contrary, microfluidics models are for sure lower cost, and also it's possible to control the microenvironment, and also it's possible to apply the shear stress and the different type of flow rate, and also apply different input of like chemical and so on. So the aim of this work is to develop a vascular tissue interface using a double channel microfluidic chip. In particular, in one channel, we cultured endothelial cells. In the other channel, we developed a 3D matrix to support endothelial cells. And we test the vascular permeability of endothelial cells against different nanosystems. And also we try to modulate the vascular permeability using a biological and also physical method using, like, for example, mannitol and Lexiscan. So the double channel microfluidic chip, as you can see, is composed by two channels that are connected in the middle part by an array of micropillar. The length of the micropillar is around 500 micron, and the gap between the micropillar is around 3 micron to guarantee the exchange of fluid and also small nanoparticles. So in the first step of our work, we focused on the extravascular compartment because we want to realize a 3D extracellular matrix in which we mixed collagen and matrigel in a different volume ratio in order to obtain a matrix that was also enriched with a different growth factor and also different type of matrix, different type of protein, like for example proteoglycans and laminin and collagen 4, that are the protein that we found in matrigel. So we mixed collagen in a different matrigel in a different volume ratio, and then we calculate via IMAGE analysis the porosity of the matrix and also the permeability of two different probe, like Dextran 250K, that is around 10 nanometer, and also microsphere of 200 nanometer. And we see that when we reach 20% of matrigel incorporated in collagen, we are still in a range in which both of the probe are able to extravasate in the extravascular compartment. But when we increase the concentration of matrigel around 30%, we are like stiffening the matrix, and so we are not able anymore to permeate dextran and microsphere. Then we focused on the vascular compartment, and we choose UVEC endothelial cells as endothelial model cells, and we grow them for 2 days. And as you can see from the picture, both in the SEM micrograph and also by confocal image, the cells adhere to the PDMS wall that are functionalized with fibronectin, and they display also V-cadherin protein junction, confirming the formation of a stable endothelial barrier. At this point, we decided to test different type of particles, and we started from 11 nanometer for like Dextran 250K through 200 nanometer bits, and also we tested 2 different platforms that we are developing in our lab, that are soft discoidal polymeric nanocastruct and also rigid discoidal polymeric nanoparticles that are around 1 micron. And so at this point, we tested the permeability of this different probe without UVEC, so just testing the extracellular matrix with the presence of UVEC, and also we tested mannitol and Lexiscan, that are 2 different modulators of the endothelial barrier. In particular, mannitol is a clinically relevant model that is used also, for example, for the osmotic opening of the endothelial barrier, of the blood-brain barrier, and also Lexiscan was used in particular for cardio application, but had also some clinical trials for the selective opening of the endothelial barrier. So we applied mannitol and Lexiscan for 30 minutes in our microfluidic chip, and as you can see from the graph, our microfluidic platform displayed size selectivity against all different probes that we tested, but in particular by applying mannitol and Lexiscan, we have an increase of the accumulation for dextran and beads, but we did not observe an increase in accumulation of soft and rigid nanoparticles that are more or less impermeable both in physiological condition that in opening of the endothelial cells. So what we can conclude from these first studies is that we had an in vitro vascular tissue interface microfluidic chip that was developed via soft lithography approach, and the vascular Biobassilonachip facilitated the characterization and the modulation of the vascular permeability, giving the possibility to predict the accumulation of nanomedicine into the extravascular compartment. Particularly, vascular permeability was tuned in order to achieve permeability value that are physiological, like the one reported in vivo, but also we showed an increase in vascular permeability by treating endothelial cells with mannitol and lexiscan. So these studies provide the evidence that vascular microfluidic platform are essential link between human physiology and animal study, and have also the potential to contribute to the development and optimization of new strategy. So all this work was possible thank you for my great lab, the Decuzzi lab at l'Istituto Italiano di Tecnologia in Genova. And this is our city and this is our lab. And thank you so much for your attention.
Wonderful. Thank you so much. That was a great talk. We have time for Q&A if people want to keep typing in their questions there. I guess I'll lead off a little bit. I really like your nanopillar design in your chip, and I was wondering if you looked at different parameters, if you varied the gap between those nanopillars, if you looked at the effect of that.
Yeah, that was a study that was done previously in my lab, and they tested the different gap size and also different shape of the, let's say, the micropillars. And at the end, they ended up with this configuration that was allowing the culture of endothelial cells, and the gap size was around, the gap size that they chose was around 3 micron, because it was the one that was allowing the accumulation of different particles in the extravascular compartment. So it was the one that was giving the better results compared to the in vivo.
Cool. Awesome. And in terms of future directions, what are directions that you're most looking forward to?
So for sure, we want to co-culture other type of cells in the extravascular compartment in order to have supporting cells that can give us the possibility to achieve more physiological in vivo value of permeability, because of course, as was mentioned also before, UVEC cells are vein endothelial cells, so they rapidly lose the phenotype. So we wanted to co-culture endothelial cells with, for example, fibroblasts or muscle cells, or tissue-specific supporting cells like pericytes and astrocytes in order to modulate the blood-brain barrier.
Awesome.
So, the action.
Cool. Awesome. And then we have a question about the protein concentrations that you use. I know you talked about varying that, but maybe some sense of concentrations of the matrigel, and then if it was reduced growth factor, any other kind of protein influences there.
Actually, the matrigel that we tested was not growth factor reduced matrigel. So for sure there were a lot of growth factor, like vascular endothelial growth factor, that were affecting, of course, also the permeability and also the organization of collagen. Because as you can see, when sorry, maybe I can show you. So when we reach, for example, 50% of collagen with 50% of matrigel, there were like a packaging of the fibril of collagen into the matrigel. So we had this kind of blocking of the chain of collagen, so a kind of effect on the polymerization of collagen fibrils. And but for sure we are also testing matrigel growth factor reduced, because we want to understand if there is an effect on the also on endothelial cells.
Great. Let me just double check Q&A. I think this is good for now. So yeah, thank you so much, and
Thank you so much.
best of luck with everything. And we'll turn it over to our next speaker, who is Laura Alderfer from the University of Notre Dame. There you are. Awesome.
And I think that looks okay on your
That
end.
looks great.
Okay, perfect. Okay, so yes, thank you to the organizers for all of their
So
hard work.
Laura, sorry, we're still seeing your presenter view.
Oh, okay, sorry. Let me.
You may have to just share that screen.
Yeah. Okay, I think that's good now.
Yes, beautiful.
Okay, perfect. So yeah, thank you to the organizers, and I'm happy to share my work with you this morning and meet some other great trainees. So our lab focuses on lymphatic vessels, and so we're trying to develop a model that we could use to study both the growth of them and also use them as a model for drug screening and disease modeling. So I just want to highlight quickly, we've already talked about it a little bit this morning in the previous speakers, but the lymphatic system, it pervades our entire body, and it's a secondary circulatory system. It has a multitude of diverse roles, but most importantly, it transports our interstitial fluid, as well as has lots of roles with immune function. It controls fat absorption, and it also clears toxins. So when we look at developmental biology for our inspiration of our model, we see, looking at, these are cardinal veins in an embryonic mouse. We see that when PROX1 is high, around embryonic stage 9.5, that we see PROX1 initiate the sprouting of the lymphatic vessels from the shared lineage. And then we see VEGF-R3, and this VEGF-C gradient is actually what causes the budding of these vessels that become the differentiated lymphatic vessels. So looking at the different shared expression of the vessels, each stage of the vessel has different macrostructures and gene expression. And so we look at the capillaries, which are the smallest. We see a higher expression of LIVE1, but then when we look at the collecting vessels, we see the development of these valves, and we also see PROX1 is higher, as well as FOXC2 and GATA2. So that just highlights the heterogeneity of a vessel, as well as the complexity of the system, which I just want to highlight as well. All these markers were discovered about 20, 25 years ago. So compared to other fields, it's pretty young, and we're trying to catch up. So we're all interested in organoids at this conference, and I just want to highlight the clinical need and hopefully convince you why lymphatics are important, and hopefully why we can incorporate them into models in the future for a more robust and a more accurate disease model. So we look at lymphedema, which most people know as one of the most common dysfunctions of the lymphatic system, and we see this chronic swelling, which affects over 250 million people worldwide. We also see that lymphatics are implicated in a host of neurological disorders, including Alzheimer's. So if we can somehow harness the lymphatic system and utilize that, we can maybe use that as a therapeutic. And then we also see an increased lymphatic response post-MI. And so can we somehow use a tissue engineered response to reduce or prevent the chronic damage? So looking at previous groups, there's been several attempts at lymphatic vessel engineering. Most commonly, they will use fibrin or collagen, but as Cody discussed, there's some inherent issues with controlling the system when you adjust the densities of those or the percent ratios. And then also we see a lot of groups will use VEGF-C, which from developmental biology, we know that's the driving force for lymphatic sprouting. And so we see increased lymphatic formation in the presence of increased VEGF-C. And then additionally, we know that LECs express LIVE1, which is also a homolog of CD44, and that'll be important for HA, which we're using in our system. So based on the markers on our LECs, we chose an HA system, and We use a PEG-DA crosslinker, which allows us to precisely tune our mechanics while keeping our other components the same. So we're keeping our gelatin and HA fixed in order to keep all of the integrins and binding sites constant while just changing the stiffness. And then interestingly, we see that the expression of our LECs is changed as a result of only the mechanics. And so when we have our softer gel, we see an increase in LIVE1 expression, which, remembering back to the other picture, that's important for our capillary development. And we also see an increase in PROX1, which is important for our collecting vessels. So this is telling us that the mechanics alone are somehow controlling our system, and we can maybe tune those to create the type of lymphatic vessels that we desire. So looking at the lymphatic vessels that we're growing on these hydrogels, this is just a representative picture of what we would see on our softer gels. And we're using this skeleton system. It's called CAV, or Kinetic Analysis of Vasculogenesis. And that was developed by collaborators at IUSM. And that's what we'll use to characterize our formation. And so when we look at the network size, which would be the area of our vessel, of the lumen, we see that as the substrate is softer, we have larger lumens forming. And additionally, as we introduce more VEGF-C, we can also have larger networks forming. And so both the biochemical and mechanical cues are having a governing effect on our system. And then additionally, when we look at the tubes per node ratio, which can be considered the extent of branching, we also see, again, that the higher VEGF-C and the lower stiffness are the most ideal. And so that, again, is telling us that there's multiple factors that are influencing our system. And then, so we can see here, this is on the softest one, which was the ideal case. We can see that there's a difference in expression of phalloidin. So on our softer gels, the LECs will tend to bind to themselves and form these junctions, whereas on our stiffer gels, the cells will tend just to bind to the substrate and not form these vessel-like structures. And then looking at TEM, we can see that as well, they're forming these vacuoles and lumens. And so that is telling us that we believe from this that, yes, they would be characteristic vessels that we'd expect to see in a mammalian model. So looking at, again, how this substrate stiffness is influencing our system, we see that, again, on the softest system, that our PROX1 expression is higher. And as well, we see that on our stiffer one, that TAZ is higher. So YAP and TAZ are mechanoreceptors, but interestingly, in vascular approaches, they're not always associated as one-to-one in their reaction, whereas other situations they tend to be. And so interestingly, we see TAZ responding to our matrix stiffness, whereas YAP is somewhat neutral. But we can see this when we look at our stiffer gels here, we can see qualitatively that there's a reduction in TAZ as we reduce the matrix stiffness. So on the left is the softest and on the right is the most firm gel. And then also we can see that the YAP, while the PCR data does not really, it doesn't completely agree with what we see in the immunofluorescence, we see this reduction in YAP expression in the nucleus. So it's important to note that our PCR data, it includes the combination of both the cytoplasmic and the nucleus expression, whereas in our immunofluorescence here, we can see where exactly YAP is localized within the cell because it has different functions, whether it's in the nucleus or in the cytoplasm. So here we see as well mechanoreceptors that are responding differently to the substrate stiffnesses. And then additionally, we looked at MMPs and to see how they were responding to the stiffness. MMPs are associated with matrix remodeling. So we looked at 1, 2, and 14. And 14 is the pro MMP for 1 and 2, as well as MMP2 is associated with gelatinases, which makes sense for our system. So we see again that on the softest gel, we see this significant difference between MMP expression. And so we're thinking that on the softer gel that the cells are secreting more of this and they're remodeling their environment quicker, enabling the lymphatic vessel formation that we've observed. that only occurs on the stiffer gel. Additionally, we see the same trend for MMP14. And when we look at FLT4, which is the receptor for VEGF-R3, or it's VEGF-R3 is the receptor for VEGF-C, we see, again, there's an increase with just stiffness alone, not even by the addition of VEGF-C. And this agrees with the previous report from another lab. And so we know that just by altering the environment, we can tune the genetic expression of our cells in hopes to create the system that we want. And then this is also the protein, but I'll quickly go through to the next. Then we also knocked out MMP14, since we know that's important based on the previous slides. And we can see as well when we knock out PROX1 that in both of these cases, you might have some alignment, but it's not the branching that we've seen before. And in the luciferase control, you can see some differences. So as Cody mentioned, we're using NORHA as well. So it's a great hydrogel, and we're happy to be collaborating with the Burdick Lab. And so we're using this to go forward into a 3D model. And it's really great because we're able to tune the stiffness, and we can also incorporate size. So as their paper beautifully shows this gradient, we're hoping to harness that for our lymphatic vessels. And so just some very preliminary studies with that, I've incorporated different amounts of RGD, which is the binding site of fibronectin. And we can see that by incorporating different concentrations of the binding site while keeping the rest of the system constant, that we can actually have differences in our vessel formation, which that was pretty promising or exciting. And so moving forward, we're hoping to see how we can tune each system of our, each parameter of our system, and then hopefully be able to train these vessels. Both, can we make collecting vessels versus capillaries? Can we form this branching that we want? Can we also introduce interstitial flow? Because as we know, vessels have flow through them, and it's not just a static culture. So if we want an accurate or a realistic model, we need to introduce our flow and see how that alters it. And then additionally, we can see from this schematic in one beautiful paper that the different subsets of lymphatics have different structures. And so we can't just make one lymphatic vessel. We need to alter, are they valves or capillaries? Are they buttons or zippers in regards to the junction? And that will be important if we want to study, say, fat absorption across the lacteals in the intestine, or if we want to study the lymphatics of the heart, or the draining meningeal lymphatics of the brain. And then also we want to move to a fibrosis model, which with the NORHA system, it's really great because we can do sequential reactions and actually stiffen the gel as our study goes on to have a dynamic system. So with that, I'd like to thank my lab. And I've had a lot of great funding sources and undergraduate students in the lab that have helped with this. And I'd like to take any questions.
Awesome, great talk. So we have a few questions from the attendees. The first is if you have evaluated or measured the stiffness of the matrices that you're making.
Yes. So we have done parallel plate rheology, and I kind of zoomed through that a little quickly. But we do see a highly linear relationship between just our PEG-DA and our stiffness. And so looking at the bulk modulus, our softest gel is around 30 pascals, and our stiffest is the 2% that we use, and it's around 300 pascals. So they're all pretty soft. You can see here that it doesn't really retain its form that much. But yes, we have done rheology on them.
Very good. And in terms of the VEGF-C that you use, did you see if using too much of it would lead to the formation of a leaky lymphatic vasculature?
That's a great question. We have not looked at that specifically, but there's been a lot of labs that have looked at VEGF-C. And so generally they look at ranges of 0 to 100 nanograms per milliliter. And we see when we got to when we're at the higher range of around 100 nanograms per milliliter, there's chronic exposure to VEGF-C can lead to issues such as the leaky membrane. We've stayed at 50 nanograms per milliliter, but we haven't measured the permeability yet.
Cool. Great. And then you showed kind of the development towards the end of your preliminary data with the RGD incorporation. Are you now thinking about looking at what you showed previously with the 2D culture across the range of stiffnesses and seeing when you incorporate these peptides as well, if those results would be affected?
Right, yes. That's another good question. And I just went through it quickly for time. So yes, we're hoping to both modify the stiffness of our matrices as well as incorporating different amounts of RGD, as well as we can also alter the amount of hyaluronic acid in our system. During the chemical synthesis, we can alter the modifications to incorporate more or less. So yes, we need to check different stiffnesses to see how the mechanics, to basically decouple the mechanics from the RGD binding sites.
Great. And there's a couple last questions that maybe you'll be able to answer in the chat afterwards. Just, I guess, the one that came in earlier was any other motivations you wanted to highlight about using HA for the hydrogel system? I think you touched a bit on this, but if there's anything else you wanted to highlight there?
Yeah, I discussed a little bit, but most importantly for the LECs, or sorry, the lymphatic endothelial cells, HA is a great system for any cell type, but for LECs, because they have this live 1 or CD44 binding site, it's basically like giving them what they want to bind to. And so it also has the ability to tune different parameters. Cody highlighted that a little bit with how different biomaterials can have some challenges, but HA, and especially the NORHA system, it seems to have the greatest potential out of all the biomaterials we've explored so far.
Awesome, great. Well, thank you again, and
thank you
to all of the speakers from our first session here.
Great, thank you everyone. I'd like to thank Dr. Stanton and Saswat Chowdhury for helping us kickstart the symposium and moderating session 1 on bioengineered vasculature. This was a great start. I just want to let you all know that we're going to take a quick break right now, and we'll be returning just before 11:20 for our second session on, sorry, I want to make sure I get this right. I believe it's fibrosis and wound healing. So we'll see you just before 11:20.